Today's episode of our investigation of microbiology in the Tortora textbook brings us to chapter 15, Microbial Mechanisms of Pathogenicity. How do the microbes cause disease? There's a picture of a Burkholderia bacterial species that we said is a problem with chronic nosocomial infections in people with cystic fibrosis. So pathogenicity means the ability to cause disease, and we've been using that word for a long time. A pathogen is a microbe that causes human disease, as opposed to all the commensals and so forth that are not disease-causing, at least not when they're in their right environment. Virulence is a measure or a statement of how severe the disease is. So virulence factors are ways that bacteria can cause disease, for example, and the more virulent the species, the more severe the disease. How the microorganism gets into the body or gets into the part of the body where it causes disease, again, sometimes we're just seeing a microbe shifting from a place where it's a normal commensal to some place where it's causing disease, is listed right here. Here's some different ways that they can get in. They vary considerably. They can cross mucous membranes sometimes. They may have a trick up their sleeve that lets them do that. They may be able to get through the skin. Very few microbes can get through the skin, but some can get through, say, hair follicles or sweat glands and occasionally cause disease. The conjunctiva of the eye is a place where inflammation can take place, more axilla. But that parenteral route, under the skin, any way it gets under the skin. It could be through an injury or a surgical wound or any number of ways to get through the skin. Most microorganisms have a preferred portal of entry. You can't just stick them in any compartment of the body and see the same kind of disease process. Again, mucous membranes. The respiratory system is the most common route. Inhalation of various viri and bacteria. You can see a list of very familiar diseases caused by inspiration or inhalation of microbes. GI tract, probably number 2. We take in a lot of microorganisms, pathogenic microorganisms included, in our diet. And most of them are killed when they are exposed to the seething cauldron of acid that is our stomach, but a few survive. And the number that survive determines whether or not disease will take place. There's usually a dose, a minimum dose of a bacteria that will cause disease. And so if you take in enough, there's going to be enough survivors through that stomach that you could cause an intestinal disease. Urinary tract, sexually transmitted infections often end up going in through the urinary tract or the reproductive tract. Urinary tract infections are pretty common also, either because of just the transfer of microbes from the colonic region into the urethra, women, or occasionally men, or by catheterization for various medical procedures, introduce microbes. Conjunctiva, it's the lining on the surface of your eye, not where the cornea is, but around it. The characteristic white-looking membrane is called the conjunctiva, and more axilla bacteria can colonize it and cause inflammation, turns very, very red. Chlamydia, trachomatis, causes a disease called trachoma, can infect the eye. Gonorrhea, Neisseria gonorrhea, can cause infections in the eye and blindness. So babies that are born in this country are automatically treated in their eyes with an antibiotic in case they were exposed to these bacteria in the vaginal canal. Excuse me. Get over something. Under the skin, any number of organisms that are opportunistic pathogens that once they get through the skin, they cause disease. Some terminology of interest. These are terms that are used to sort of quantify the severity of the infections caused by various organisms. So The ID50 is the amount or dose of the microorganism needed to cause disease in 50% of the population. Just gives us a handle on approximately for any given individual. You can't directly translate that, but how much of a dose would be required to cause disease. The LD50 is the lethal dose in 50% of the population. Oftentimes when we're talking about LD50s, we're talking about experimental animals, but sometimes it could be listed for people too. Certain toxins are very, very lethal, and we'll have an LD50 listing for those. Here's a table of some ID50s, infectious dosed, portals of entry, endospores. 10 to 50 endospores is all it takes to cause a skin infection, but that's not such a big problem compared to inhalation of endospores. 10 to 20,000 endospores sounds like a lot, but when you have a very fine powder of endospores containing millions and millions of spores, which might get into the air around you, will easily inspire well above that number, and it could be lethal. Ingestion, a lot of endospores it takes to cause a lethal gastrointestinal infestation. LD50s, actually these are IDs, I was thinking LDs, so these are the numbers that are required to cause disease, and the LD50s for certain toxins given here. Botulinum toxin, you can see, is an extremely poisonous substance. 0.03 nanograms per kilogram of body weight is the lethal amount of botulinum toxin. So you can imagine the amount that's being injected when you go to when someone goes to a cosmetic clinic is very, very small indeed. Shiga toxin listed there. And staphylococcal enterotoxin, when you have food that gets contaminated by staph bacteria and it's allowed to grow sufficiently to produce at least 1,350 nanograms per kilogram of enterotoxin, that could be lethal. So, of course, normally when we get sick from staph toxicity or intoxication from our foods, much smaller amounts than that. Microorganisms often have some mechanisms for adhering. The first step in causing disease is usually being able to adhere to some tissue or some particular host cell. And glycocalyx, the polysaccharide covering on some bacteria, can allow them to attach to their host cells. Some have fimbria, E. coli, that can cause disease, can attach to the epithelium of the intestine via fimbria. Streptococcus pyogenes causes a whole raft of different diseases, has one of its virulence factors called the M protein, which can allow it to attach to cells. It also allows it to avoid phagocytosis, all kinds of things. Some microorganisms form biofilms, which of course allows them to stick like glue onto whatever surface the sticky biofilms. Oh, the hounds are restless. Here's a picture of a pathogen with adhesins, ligands that attach to specific receptors on a cell surface, for example, and allow them to attach on there. They may be taken up by the cell by endocytosis after that attachment takes place. They must find a complementary surface receptor to bind to. You can see some E. coli here also that are causing a UTI, a urinary tract infection, attaching to bladder epithelial cells. Here you can see Staphylococcus most likely attaching to human skin. There's bacteria all over your skin. One of the most prevalent species are Staphylococcus species because they can stand that salty, dry environment. They can survive on the surface of your skin. Hence the mannitol salt plates for selective culturing. Some microorganisms produce capsules, and capsules oftentimes allow bacteria to escape phagocytosis, and so they can't be as readily killed by the immune system. We already talked about Streptococcus pneumoniae. You take away the gene that allows them to make the capsule, they're harmless, whereas with the capsule they can cause life-threatening pneumonia, and that was the way that transformation of bacteria was discovered by the experiment where they mixed dead Streptococcus pneumoniae with the capsule gene, with live Streptococcus pneumoniae without the capsule gene, and found that they produced a lethal combo because of transformation, and the live cells acquired the gene for the capsule. Haemophilus influenza, a bacterium, also has a capsule that helps it escape or evade phagocytosis. Then Bacillus anthracis causes anthrax, and one of the most famous things that we learned about in the history of microbiology. Talked about strep pyogenes just now. There's this M protein that can help it attach to host cells. It also can help it resist phagocytosis. It interferes with opsonization, phagocytosis. The OPA protein of Neisseria gonorrhea inhibits helper T cells, so the adaptive immune response has a hard time getting going against these things. Mycobacterium species have that waxy mycolic acid cell wall, and that makes it hard for the lysosomal enzymes to digest them. And in fact, they can survive in macrophages. Mycobacterium tuberculosis causes a massive macrophages to accumulate in part of the lung, where these things are multiplying right in the macrophages, and eventually the body, in an effect to try to isolate this problem, since it can't complete it, will just becomes a hungry. Oh yeah, so the body will then attempt to wall off the problem by creating a connective tissue capsule and producing what's called a tubercle around this massive Mycobacterium tuberculosis bacteria-laden macrophages, and hold them at bay for a while. And there's this latency period in the disease of tuberculosis. A lot of microbes have as virulence factors enzymes that they produce. Coagulase enzymes cause blood clots to form. Kinases are the opposite. They cause dissolution of blood clots, and that actually allows bacteria to escape from an area of injury, liquefy the blood clot, and then be carried away in the fluids around there and move to different parts of the body, different tissues. Hyaluronidase and collagenase are enzymes that break down the structural extracellular matrix proteins of tissues. So that allows the microbes to kind of penetrate into tissues and pass on in and through by breaking down the structure, just kind of liquefying some of the structural proteins of the tissue. Some microbes produce proteases specific for immunoglobulins, antibodies. Immunoglobulin A is the antibody class that's found in secretions like tears and semen and mucus and things like that. So these bugs can break that down to prevent them from being attacked. Here's an interesting application. Here's a picture of a coronary artery tree using a contrast medium. It's been injected right into a coronary artery, and a photograph taken of it, x-ray taken of it. And you can see a blockage of an artery in someone who's having a heart attack because a atherosclerotic plaque has ruptured and a blood clot formed in that coronary artery, blocking off downstream flow. And actually, in this case, probably this is maybe not actually during the heart attack, but it's pretty bad occlusion. Once a heart attack took place, the downstream vessel would also be blocked in terms of blood flow. Anyway, a treatment that's used immediately when someone has a heart attack as soon as possible is given something to break down that blood clot that's in that occluded area of the coronary arteries. Streptokinase and staphylokinase are recombinant versions of kinases that are produced by bacteria, so they've been harnessed for medical purposes in this case. So yes, streptokinase activates the enzyme plasminogen, which is the body's normal enzyme that breaks down clots eventually, so that wound healing can progress from a later stage of the aftermath of injury. Plasminogen activates plasmin. Plasmin starts dissolving fibrin. Fibrin is the substance of the clot, so streptokinase helps break down these clots. Necrotizing fasciitis is a terrible, terrible disease with a very high mortality rate. It's when these Streptococcus species, for example, if they get into the fascia between the skin and the muscle tissue below, there's really not much vasculature in there, so there's not much access by the immune system. And if someone is already in pretty bad shape in terms of disease progression in their body, the bacteria can then travel relatively quickly in that space between the skin and the underlying tissues, the fascia. You have this necrotizing, means death-causing fasciitis, inflammation of the fascia. And there's a picture of very rapidly moving bacteria that are causing this so-called flesh-eating disease or flesh-eating bacteria. Other bacteria can cause a similar result if they have the right enzymes to be able to penetrate in there and move quickly through. Very dire situation indeed. Salmonella typhi has an interesting virulence mechanism or disease-causing mechanism in which it can induce ruffling and endocytosis. So it has a special mechanism of injecting bacterial toxins, exotoxins, into the host cell, into the epithelial lining of the digestive tract, and that causes the cell to change its actin cytoskeleton in such a way that it produces this ruffling effect and induces endocytosis of the bacteria. Now they have entered into the cells and gotten to the, actually maybe it's the M cells of the wall. I'll have to look that up, the wall of the intestine. Now they've gotten inside and they can survive in there and cause a transfer to the other side and cause disease, enter the bloodstream ultimately, and cause systemic disease. Another way that some microorganisms can cause disease, another mechanism, is to shift the antigen on the surface of the microorganism. These antigenic variations. So the antigens on the surface of the microorganism or the parasite will shift just as the immune system. Remember how long we said it takes for an adaptive immune response? Probably a good antibody titer is going to be coming along right around a week after infection. Well, that's right about the time that the microbe, here in this case a trypanosome, right around the time that the adaptive immune response is really gearing up, the microorganism or parasite changes the antigens on its surface, and now all of a sudden they're brand new again. The antibodies that were just produced are useless, and so on and so forth. So this, a couple of different microorganisms have this antigenic variation capacity, not just trypanosomes. Influenza. Is that right? Invasins. Some microorganisms produce invasins. We just talked about salmonella, how it can enter a host cell by injecting some proteins that cause endocytosis. Interestingly, Listeria bacteria can also get into epithelial cells. Then the Listeria can control the actin filaments polymerization and the shape of the actin cytoskeleton, and so that it can be pushed from one cell right into the neighboring cell, so that it can go from cell to cell and multiply without ever coming out of the cell and becoming visible to the immune system. So it'll trick the Listeria bacteria produced to cause disease. And here's a picture of an latent type of a disease in which the infection is being manifest here by a human herpes virus type 1, presumably, which has been lying dormant in a neuron, and now it has for some reason been activated. Probably the person became overtired or stressed, and for some reason that caused activation. Out comes a virus causing another lesion in the body. Another way that microorganisms can get a foothold or get a leg up on causing disease in the body is by getting a hold of iron. All living cells need iron for various enzymes that they need to survive, and iron is kept under pretty tight wraps in your body. It's always bound tightly to a carrier protein, partly because iron is nasty, toxic stuff. Otherwise, it goes around reacting with things, and so it's kept bound to carrier proteins, and it's kept in the bone marrow and the liver, and if it's in the bloodstream on its way somewhere, it's all bound up by transferrin and other binding proteins. But some microbes produce siderophores, which are chemicals that can grab that iron, have even a higher affinity for the iron than the host's binding proteins, and so they can grab that iron once they have an access to iron, they're in business as far as being able to multiply and do what they do. All kinds of other things that microbes can do. Um, they can uh, disrupt host cell function, maybe by waste products or toxins. All kinds of toxins that are cell specific, as we'll learn in a little bit. They can cause fever, cause cardiovascular problems. Some microbes cause inhibition of the cardiovascular system when they get into the blood. Some of their toxins. Diarrhea. We talked already briefly in a few occasions about bacteria that cause diarrheal disease or protozoa or even helminths, mostly protozoa and bacteria. Shock, I mentioned about shock. Some toxins can cause a drop in blood pressure that's so severe it can be lethal. Um, Inhibition of protein synthesis, a way to kill cells, stop their protein synthesis. They can't survive without being able to have protein turnover and constantly removing proteins in the cell. Inhibition of metabolism, surely if the cells can't produce enough energy, they can't survive. And damage to cell membranes, quite a common mechanism of toxins. So toxins, all kinds of toxins that meet all those criteria or work in all those different ways. Toxins are substances produced by microorganisms that causes disease. Toxicity is the ability to produce a toxin. Toxemia means the toxin has now arrived in the blood. Remember that suffix emio you've seen many times. So now you have toxins in the blood. A toxoid, interestingly, as you know now from studying the immune system last chapter, is a denatured or inactivated toxin. So when we take a toxin that causes severe disease and we want to make antibodies against it, we can inject some denatured toxins into the body and prevent and produce antibodies and memory cells to prevent that toxin from ever being life-threatening danger to the body. Antitoxins or antibodies against particular toxins, that's what's produced when we inject a toxoid vaccine. Antitoxins will be produced, and if the infection ever arrives in the body of the bacteria that produces that toxin, I meant to say, then it won't be able to cause disease. So, toxins. A little bit more about toxins. Exotoxins are produced by bacteria that are secreted. They go out of the bacterial cell and cause disease either by direct secretion into the surrounding fluids, or some exotoxins are injected. Some bacteria have a really cool thing where they can actually inject, like with a little syringe, toxins into a cell. They bind to the surface of the cell and they inject the toxins. Gram-positive bacteria are the major exotoxins producers, but gram-negative bacteria produce some very notable toxins as well. Endotoxin, as you know already, is a component of the gram-negative bacterial cell wall. The outer membrane, the outer leaflet of that membrane is made up of lipopolysaccharide. The lipid A component is called endotoxin. It's highly inflammatory to mammalian cells, including human cells. Exotoxins tend to be more toxic in terms of the LD50 than endotoxin, much more so. Some exotoxins are just exquisitely lethal chemicals. Exotoxins are often named for the cell type that they attack. Neurotoxins obviously damage or inhibit functions of the nervous system. Cardiotoxins on the heart. Hepato means of the liver, so hepatotoxins and leukotoxins. Leukotoxins attack leukocytes, white blood cells. They prevent immune function in defense of these organisms. Sometimes exotoxins are named for the disease diphtheria. Corynebacterium diphtheriae produce a disease a very horrible disease called diphtheria, in which these microorganisms produce a toxin that kills the epithelial cells in the pharynx and cause tremendous amount of swelling, and that it can actually cause suffocation. Tetanus toxin, we learned about that. Mycobacterium tetani produces this tetanus toxin that can cause paralysis, spastic paralysis of skeletal muscles. And by bacterium, botulinum toxin is named for the bacteria. What did I just say? I think I just said that wrong about tetanus. The mental block. Clostridium, tetany, Clostridium botulinum. I think I said something else. I think I said Mycobacterium. Sorry about that. So yeah, Clostridium tetany produce botulinum toxin, and so that's the name of the toxin. One type of toxin, just for fun, to enumerate the mechanisms of action a little bit more. AB toxins are the most well-studied bacterial toxin class. They're like a two-part toxin. They have one part that binds to a receptor on the cell, on the host cell membrane. That causes endocytosis. They go into the cell, and then they separate into the A and B component, and then the A component is what actually causes toxicity to the host cell. So if we have a leukotoxin, that's going to be getting into the cell, and that A component is going to be what kills that cell, stops it from doing protein synthesis or something like that to kill the cell. A lot of toxins, we saw about 40% of toxins cause interference with the cell membrane. Leukocidins, hemolysins, and streptolysins are all toxins that cause problems by damaging the cell membrane and killing cells that way. Again, leukocidins kill white blood cells. Hemolysins cause lysis of red blood cells in the circulation, disrupting the phospholipid bilayer, killing cells that way. Superantigen, here's an interesting topic. Some toxins, in addition to maybe causing one problem, also can be superantigens, or just maybe they're not even separately a toxin, but superantigens are chemicals that can cause non-specific activation of T cells. We said that helper T cells produce cytokines, which can promote inflammation but activate cytotoxic T cells and B cells. Well, when you have a superantigen, it goes around to virtually any and all helper T cells in the area and turns them on, and it produces a massive amount of cytokines, and you can have a lethal inflammatory response, or at least a very vigorous inflammatory response. So shock, what it's called when the blood pressure falls so drastically that there's not enough circulation to meet the needs of the body tissues. So that's one of the things that happens when you have a superantigen response. You have so many cytokines causes vasodilation. Inflammatory chemicals are released, vasodilation, blood pressure tanks, and you can have shock and death. All right, we've already talked a little bit about exotoxins. Low LD50s, it doesn't take very much of a typical exotoxin to be lethal to the body compared to gram-positive or gram-negative bacterial endotoxins. Takes a lot more, but that's not a problem for them. They can use a lot more because, remember, it's part of the cell membrane, cell wall, I should say. And so if you have an ongoing infection with gram-negative bacteria, there's plenty of endotoxin to go around. A lot of exotoxins are produced by lysogenic conversion. We talked about the ability of some phage genomes to integrate into the bacterial chromosome, and then when they come back out again, when the lytic cycle restarts, they may take with them some genes, and a lot of the genes that they take with them are exotoxin producing genes, so they can convert a harmless bacterium into a pathogen by introducing a exotoxin capability into those bacterial cells. So you can see a bunch of toxins that are transmitted horizontally by phage bacteria this way. Again, endotoxin, very large LD50. It takes a lot more endotoxin to kill, but yet there's a lot of bacteria presumably in the infection, so it can be produced that way. Endotoxins cause fever. Endotoxins wind up getting into the bloodstream. They're released from gram-negative cells when they die, right? If you treat somebody with some antibiotics and they have a gram-negative infection, it's a very risky thing to do, because then a lot of endotoxins released from the cell wall. Or when cells divide, you tend to release a fair amount of endotoxin. Anyway, if endotoxin gets into the blood, then it can go to the hypothalamus prostaglandins will be synthesized in response to these endotoxins, this inflammatory capability. And then one of the things that will happen is hypothalamus will upregulate the body temperature, and that's what we call fever. We have a new regulated value for body temperature that's higher. Here's something very fascinating. How do you know if endotoxin is present in a solution or a medicine or something? When medicines are made, especially ones that are made from body fluids or from animals, we have to be very, very careful to make sure there's no endotoxin in even a trace amount in the medicine that's produced at the end. So we need a way to test for that. And the current sensitive endotoxin test, strange as it may sound, is to take some horseshoe crab blood. Yep, those horseshoe crabs that are found in the rivers emptying out into the ocean around Athens or around Savannah, Georgia, bazillions of horseshoe crabs down there. Well, yes, take their blood, and they have some cells called amoebocytes in the blood, which when lysed, you can break them open and produce a preparation that when exposed to very small amounts of endotoxin will clot. It'll gel right up. And so that's a way. Taking assay kits made from horseshoe crab blood is the way that endotoxin is typically tested. It's a big problem in a sense because there's only so many horseshoe crabs, and this is a big industrial thing now to take thousands of horseshoe crabs and take all their blood out and use it for making this substance. But now the efforts are being made to farm horseshoe crabs, which initially was not possible. I don't know how far they've gotten this yet, but so that they can, so a sustainable production of horseshoe crab blood can be realized for making these test kits. Viruses produce a cytopathic effect. They change the cell function. When they get inside of cells, they cause all kinds of changes in cells, and that can greatly affect the performance or the function of tissues. So viruses cause all kinds of pathogenicity based on those changes that take place in cells. Some viruses actually can cause cancer, and here's a picture of some transformed cells in a sample of tissue that's been infected with a virus, and now we have some tumor cells right there in that picture. Fungi, we're tapering down now, funneling down a little bit. Fungi produce some toxins of great fame and notoriety, but not very often. We are pretty careful not to allow these things to get too much of a foothold in our agricultural process. Usually fungal toxins are produced not in the body but in the agricultural productions, and so people are exposed to fungi, for example, that get into various grains that are then used for food production and so forth, and you can have some toxins. Some fungi can produce toxins. Trichothecene toxins inhibit protein synthesis and kill cells. Some bacteria produce proteases that can allow them to overcome tissue limitations and so forth, get into the skin, for example, and start causing infections or epithelia. Cryptococcus neoformans, and I forgot what the other disease-causing species. Cryptococcus is a fungus that's associated with decaying bird droppings in the inner city, pigeon droppings, which can cause a type of pneumonia in immunocompromised subjects, and the way they cause disease is by producing this capsule that protects them from phagocytosis. So, some famous toxins produced by some fungi. Ergotoxin is produced by claviceps and some other fungi that can contaminate rye grains and actually some other types of bread grains and produce a toxicity. Gastroenteritis, familiar syndrome, we already know about from a lot of other things, but at higher doses can actually cause a severe burning sensation in the skin of the limbs. That's a famous name for that condition. It's called St. Anthony's fire. Aflatoxin is a fairly powerful toxin that's produced by growth of various aspergillus species of decaying vegetation in the soil. It can cause liver damage, kills liver cells. This homes in on liver cells and causes liver cell death. Um, um, Mushrooms in the Amanita family, I probably should have capitalized that. What was I thinking? Amanita mushrooms carry some mycotoxins. Phalloidin and amanitin are very, very potent toxins produced uh, by members of this family. Phalloidin causes liver toxicity, I meant to say. The other one causes death, very, very powerful toxin from the so-called death cap mushroom of this family. But the reason I included this little description is kind of cool that scientists are always trying to collaborate with people from different fields and coming up with ideas like taking antibodies and attaching toxic payloads, they're called, to them, so that they can home in on specific types of cells, such as a cancer cell type, and then bring a toxic substance to those cancer cells, such as um, this this toxin, this very powerful toxin from the death cap mushrooms. Protozoa have all kinds of disease-causing capabilities. Just their presence can cause problems. They can cause physical problems. They can fill up your entire GI tract with volumes of worms. Um, their waste products can cause disease. Trypanosome produce all kinds of secretions or stuff that's released when they die that can cause irritation and disease, inflammation, and so forth. Some microorganisms can avoid host defense by actually being able to grow in phagocytes. Leishmania and toxoplasma causing parasites can live in phagocytes and survive and grow. Antigenic variation we mentioned before with, we saw a little graph of trypanosomes making different antigens to place on their surfaces and rotating in order to escape from the attacks by the adaptive immune responses, particularly the B cells and plasma cells and antibody production. The Plasmodium, the Babesia species, and Giardia species can also exhibit antigenic variations. So that's how they can kind of keep ahead of the immune system, and so they're hard to kill, can have a chance, therefore, to multiply and cause disease. Some helminths can actually eat host tissue, like liver flukes can actually eat liver tissue, and they also can cause overgrowth of cells, some of the cells in response. A couple of different parasites do that. Apparently the bile ducts in the liver can hypertrophy and overgrow and cause blockage by the action of those flukes in the liver. Encystment of different helminth types can cause tissue damage. Sometimes it just causes, they just enlarge and cause the cells kind of die around them by kind of blockage. Blockage of the intestine by ascaris worms and tapeworms. That's another type of issue, pathogenicity with those guys. Normally they don't cause too much trouble. They just eat up all your nutrients, but they can actually cause other problems as well just by their very quantities. Some waste products produced by parasites can cause disease. Paralytic shellfish poisoning. Dinoflagellates, we already talked about, can cause the so-called red tide, in which they contaminate fish, which are then eaten by people and made sick. Those are neurotoxins. Also, saxitoxin is a terrible neurotoxin produced by certain bacteria that can then accumulate in freshwater fish and can cause lethal intoxication. There's a picture of the red tide that we actually saw once before of dinoflagellates that grow in the water at a very high confluence, therefore producing toxins. All right, so portals of exit, our last little topic, I believe. How do microorganisms go out of the body? We talked about portals of entry. Well, portals of exit are important to know about too, because that's how often disease is transmitted from one organism to another, one individual to another. Coughing and sneezing, so from the respiratory tract. Feces, fecal-oral transmission is hugely important. Many diseases. Salivary transmission can occur through the gastrointestinal system. Urogenital tract, urine and vaginal secretions can carry pathogens from one host to another. Sexually transmitted infections and so forth. Skin, blood, arthropods that bite can pick up microbes from the blood and then transfer to another host. People that are injecting drug addicts can transmit disease by sharing of needles and syringes. A lot of governments are trying to get around this problem by providing free sterile syringes and needles in areas where there's a lot of drug abuse happening. When people are addicted, they can't stop themselves, and you might as well keep them from having comorbidities, other diseases other than their addictions to deal with in their lives. So, summary slide here, portals of entry, talking about some of the ways that microorganisms can get into the disease, how they can get in and evade the host defenses, cause damage to host cells, and portals of exit. You can look at this slide and make sure you can give some examples maybe of a few of these different processes. But at least have something to say about those from our chapter. So that's it for chapter 15, did we just do? And we'll be back again soon. I believe our next chapter is on antibiotics. So join me for that.